Okay, Bio 30. So we're now going to start the next section, which is a continuation of the nervous system. But this series of videos is going to be about how the nervous system propagates information along a neuron. So to begin, we have to discuss some sort of basic information vital to understanding what an action potential is. Like, what is the transmission of an electrical impulse along a neuron supposed to be? What does it look like? So resting membrane potential. So remember, we're dealing with electrical charges here. And so when we have a neuron, it has a charge, and we call that the resting, well, we call it a membrane potential. So at rest, when a neuron is not transmitting any signal, it's just sitting there waiting for a signal to come, its charge is negative 70 millivolts. So volts is a way of measuring electrical charge. Millivolts is a unit, and then negative 70. So it's got a negative charge of 70 millivolts. This is when the neuron is not active, when it's at rest. So an unstimulated axon or neuron is negative on the inside in respect to the outside. So we're going to keep looking at a neuron. Imagine a tube. And inside that neuron at rest, it's got a charge that's negative in comparison to the outside of that neuron. So here's the neuron I'm kind of trying to talk about here. It's unstimulated. We've got the outside and we've got the inside of that neuron or axon. Outside the neuron at rest, it's positively charged in comparison to the inside of that neuron, which is negatively charged. And so what we say is the membrane potential, or the difference between the outside and the inside at rest, is negative 70 millivolts. An action potential is when the neuron is no longer at rest. It's been triggered. There is a signal being propagated throughout that axon or throughout that neuron. So when the axon becomes excited and is actually transmitting an electrical impulse, there is a charge difference across the membrane and it changes. So now, in a stimulated axon, the outside is still positive, the inside is negative, but I'm going to show you an action shot here. So this is where this might get a little confusing, but I'll kind of try and work it with you here. The outside at rest should be positive. The inside at rest should be negative. But as soon as an action potential occurs and that signal is propagated or moves along an axon, the inside becomes positive and the outside becomes negative. Okay, so now this is where you have to bear with me. Imagine this electrical impulse moving. So it started over here where this was negative on the outside and positive on the inside. And then it moved over here where this went back to a resting state, positive on the outside, negative on the inside. And now this portion of the membrane of the axon is now stimulated. So now it has become negative on the outside, positive on the inside. And then this portion is yet to become stimulated because the action potential hasn't reached that location yet. So this is still positive on the outside and negative on the inside. And so it'll just keep going. This action potential will keep moving along the axon, along the length of the axon, as that action potential moves throughout the neuron or the axon. So the action potential switches to a positive 40 millivolts. So at this particular location where the action potential is traveling is now a positive 40 millivolt charge difference, versus before at rest it was negative 70 millivolts. So we've actually become quite positive on the inside compared to the outside. Now we will break down the anatomy of why it's negative on the outside and positive on the inside during an action potential in a lot of detail in a second. Okay, so right there is our action potential, and it is moving or being propagated down the length of the axon. Okay, so resting neuron, what's creating that charge difference? Well, the charge difference is due to positive ions. So this is something very critical to understanding. There's going to be there's more, but we're going to focus on two major ions that are relevant to making a signal get transmitted down a neuron. Sodium ions, which are positively charged, and potassium ions, which are also positively charged. So Na and K, Na plus and K plus. There are more sodium ions outside the neuron at rest than potassium ions inside the neuron at rest. Okay. So let me just kind of show you with a picture. It's a little bit easier here. So notice how there's far more sodium outside the neuron than inside the neuron. And here's our neuronal membrane. And notice that there's a lot of channels and gates here. Um, so sodium at rest exists in a much higher concentration outside the neuron. Potassium at rest exists at a much higher concentration inside the neuron. And there's very little sodium in here. Now, I always like to remember this as the king always lives inside of his castle. So at rest, when things are safe and things are good, you're always going to find the king, K, inside of his castle. So potassium at rest is always in a higher concentration inside, and sodium is always at a higher concentration outside the neuron. Okay, so what this is going to do is we are going to here, I'm just going to back this up here for a second. Since there's more sodium ions on the outside and there's more potassium on the inside, when there's potassium on the inside, there also happens to be these negatively charged proteins that sort of accompany the potassium. So what this does, even though sodium is positive and potassium is positive, these negatively charged proteins, which we won't get into the details of here, but these negatively charged proteins are sort of hanging out with the potassiums. And so wherever potassium goes, those negatively charged proteins are going to go. And that means that the inside is going to be more negative relative to the outside. And this is why you have that negative 70 millivolts. The inside of the neuron at rest is normally much more negative than the outside. So the charge difference here is minus 70 millivolts. Now, during an action potential, sodium channels are going to open, and that's going to allow sodium from the outside to diffuse inside the membrane. So there's the sodium coming in. Now, as the sodium concentration increases inside of the axon or inside of the neuron, the charge difference begins to decrease. We start becoming more positive than negative 70 millivolts. So if it was negative 70 millivolts resting membrane potential at rest, then as the sodium rushes in during an action potential, during a signal being transmitted, you're going to have more or a higher increase in positivity of that membrane potential. So instead of being negative 70, it's now negative 65, and now it's negative 55, and now it's negative 40, and now it might be plus 10. And then action potential won't actually occur, or a full signal won't propagate down the length of that axon until it reaches positive 40 millivolts, which is the critical point where you need to have a membrane potential occur or a signal actually get transmitted. So sodium keeps rushing in, the membrane potential keeps increasing in positivity, and in addition to that, to help make the inside more positive than the outside, so to help the membrane potential become a plus 40 millivolts, not only did the sodium rush in, but the potassium rushes out. So sodium is going to make the inside more positive, and then the potassium is going to leave to make the inside more positive. And so potassium channels open and potassium, K, moves out. And so we'll kind of break this down more specifically in the next video with how that membrane potential actually occurs in a step-by-step fashion. But basically, there's sodium channels that let sodium in. There's potassium channels that let sodium out. This is going to help increase the positivity of our membrane potential to that positive 40 millivolts required for an action potential to occur. And at the very end, when it's all said and done and the membrane, that portion of the membrane wants to go back to rest, we have to put everything back where it was. So now this active transport sodium-potassium pump, which actually requires ATP because it's active transport and it's moving these things against their concentration gradients, is going to push 3 sodium out for every 2 sodium it brings back in. And now why is it an active pump? Well, it requires ATP because remember, if we're trying to move sodium out, there's more sodium on the outside than on the inside. And so we're moving sodium against its concentration gradient. Right now, when this pump works, it's going to be moving sodium from an area of low concentration to an area of high concentration. And so the sodium is going to move against its concentration gradient. It's going to require energy. The potassium, same thing. It's moving back into the inside of the membrane from an area of low concentration outside to a relatively high concentration on the inside. So it's moving against its concentration gradient. It's also going to require energy. So this ATP pump, the sodium-potassium pump, is going to carry 3 sodium in for every 2 potassium it kicks back out. And it's trying to reestablish resting membrane negative 70 millivolts after a membrane has become polarized due to an action potential traveling through it. Okay, so there you go. There's a little image of our sodium coming in and our sodium going out and our potassium coming in. All right, so another picture here just to sort of describe where things can move. I'll let you pause it here if you want, just to keep an eye on like what's what using that legend. So sodium channels are going to allow sodium to diffuse in. Potassium channels allow potassium to diffuse out of the membrane, and then the sodium-potassium pump exchanges them in their proper concentrations at the end of the membrane action potential event. So here's a little bit more information on the sodium-potassium pump. Right, it's active transport. It carries 3 sodium out. For every 2 sodium it brings back in to the membrane when it's trying to bring the membrane back to rest. So this means that there are more positive charges found on the outside than on the inside. So why is the inside more negative at rest compared to the outside? Even though potassium is positive and sodium is positive, well, notice how even the pump carries more sodium outside than it does potassium inside. So there's just always more sodium, which is positively charged, than there are potassium, which is positively charged, but there's less potassium on the inside than there are sodium on the outside, so the inside is going to be more negative or less positive in relation to the outside of the neuron, which is why we say at rest there's a negative 70 millivolt difference of charge between the inside and the outside of the membrane of the neuron. All right, so there's another little image here sequentially of how our sodium-potassium pump works. And the key thing here is to remember that it's 3 sodium gets pumped out and 2 sodium gets pumped back in at the end of the membrane potential. And this is to recreate that membrane gradient where the inside is more negative compared to the outside. We can go back to the negative 70 millivolts. Okay, so the results here before we end the video. There's a negative 70 millivolt charge inside the axon at rest because the inside is more negative compared to the outside. And so in the next video now, I'm going to describe to you what that action potential looks like and how the sodium pump, the potassium pump, and the sodium-potassium pump, or sorry, I should say sodium channel, potassium channel, and then the sodium-potassium active transport pump all work together to help allow an action potential or an actual signal to get transmitted down an axon. All right, thanks guys.